experiment is the key part to how you figure it out. So if what you're doing here tonight at home doesn't work exactly as mine does, science comes into play when you try to figure out why it didn't work. So this is something you can do repeatedly. Fiddle around with it if it doesn't work. Or if it does work, try it with something different. That's a real experiment. Doing something you don't know what's going to happen and trying to figure out what the results are and why. So we're going to get into the DNA extraction right now. And I'm going to bring you up a little photograph, a picture of what we're after. A picture of a DNA molecule right here. And DNA is actually a very, very big molecule. And it's a chain of smaller molecules. There are 4 small molecules that make up every DNA molecule. And the order in which those 4 small molecules are stuck together encodes the instructions to make every living thing. If you could take a strand of DNA, which we're about to do, out of your body's cells, each strand would be about this long. Now the reason it fits into your cell is because it's even thinner than a human hair. And each DNA molecule is twisted, as you can probably see in the picture. It's twisted around another DNA molecule in a sort of spiral. And then after they twist together in that spiral, they twist together again on each other, and then they wrap around things called histones, which are little proteins. We're actually going to be pulling some of those out tonight too. So at the end, you have this long molecule that's almost 10 feet long that's actually packed up in this really tight spiral formation with another spiral on top of that, and then it's wound up around these proteins, so you get kind of a wad. We're going to take a look through the microscope right now at some vegetable cells. And these vegetable cells, these are actually from onions. And in the middle of, this is a cell here, and in the center of each cell you'll see a dark circle. That is the nucleus of the cell. That is where the DNA is. And in some of these cells you can actually see what are called chromosomes. And these are DNA molecules that are so tightly twisted together that you can see them through the microscope. Now when they're unwrapped, they just kind of look like a purple blob. When they're twisted up, they look like a chromosome, and that's the X or Y that sometimes people refer to when they talk about chromosomes and genetics. We aren't going to see that tonight. What we are going to see are long, skinny fibers is going to be what we result with. It's just going to be the DNA from either our banana or our strawberry. So the first step to this is you're going to want to get your Ziploc bag set up. Just open the thing up and either take your strawberry or your banana, pluck the little green leafy things off the top of the strawberry, the petiole, put that in your bag and give it a good crush. So the first step is we're going to separate all these cells from each other. Now the cells are too tiny to actually break up with our hands. You can't smash the cells themselves. You might get lucky and smash a few, but we can separate one cell from another. So you want to get this into this sort of strawberry mush paste. I'm going to get that set up with the banana over here as well. Once you have that sort of thing going on, now we have just this sort of sea of fruit or vegetable cells inside these bags. And this is where we're going to bring chemistry into the equation. If you look back over here at our microscope image, we've got to get rid of this wall that's around each cell. We call that the cell membrane or the cell wall, and that's made of molecules called lipids. They're the same kind of molecules that makes up oil or fats or waxes. And that's what surrounds every single cell and keeps them separate from the outside world. So we need to get rid of those lipids. We need to get rid of all this stuff that's around the nucleus in order to get our DNA free. That's when our first ingredients are going to come in here. So you can either use a measuring teaspoon or just a regular old teaspoon out of your silverware drawer here. And we're going to use dish detergent. And the reason we use dish detergent is if you've ever seen a commercial for dish detergent, it's great at removing grease, grime, and oils. Well, the outside membrane of our cell is chemically very similar to oils. So you're going to want to put 2 teaspoons of liquid dish soap into your bag. And again, it doesn't have to be a measuring teaspoon. What's important here is the ratio of soap to water to salt, where we eventually get. So as long as you put 2 parts liquid soap in there, you should be good. Now, to your 2-part soap, you're going to want to add 1 part tap water. And I have my tap water in a beaker here, but for the ensuing steps, you can use a cup, a tumbler, plastic cup, paper cup, a glass, anything you have around the house and you have easy access to. Now we've got our water and our dish soap inside the bag here. And this mixture of water and dish soap is immediately going to start dissolving the outsides of each of these cells. Now, for all of the people out there that have been involved in outside the box labs programming at their school in the past, and you could probably help explain this to your parents, when things dissolve, it's because some molecules actually behave like little tiny magnets. They're charged positively or negatively, and those charges attract other molecules that are charged oppositely, so positive attracts negative, negative attracts positive. If you've gone through that with us, go ahead and explain that to your the people around you as we move into our next step, which is you want to flatten your bag out. My bag is still open, which is the key here. You kind of want to squeeze the air out without squeezing all of your good DNA soup, and then zip it closed. Now for the next couple minutes, we're just going to mix and mash this, and this is kind of the fun part. You take out a little stress on this here. So knead it, smash it, crush it. You don't want to pop the bag. That's very important right now. Don't break your bag, and that's another reason that we've used strawberries and bananas. They're nice and soft. They're really easy to manually crush up. And while we're doing this, not only are we breaking the cells apart from each other, but these chemicals are going to work breaking open the individual cells. And that is what is going to release the DNA from each cell and allow us to extract it. Now once you've got this a little bit crushed up, I want you to open it back up again. And I want you to take the table salt that you have. Now this is not a very scientific measurement we're about to use. You're going to add a pinch of salt. So just a tiny little bit of salt. Open up, sprinkle that in your bag, squeeze the air back out, zip it closed again, and then resume mixing up. Now we've added the salt because salt is another one of those molecules that's polar. It's charged. It's got positive and negative charges, and that's actually going to protect the DNA from the soap. DNA isn't really stable as a molecule, especially when it's outside of a cell. So as the DNA starts to move out of our cells, that soap can actually start to attack it and break it down as well. So that salt is in there. It's going to latch onto the DNA molecules and kind of protect them from the environment inside this bag. Now this is kind of cool right now, given the circumstances with the virus that we're all trying to avoid and trying to contain. Everyone's first line of defense here is washing your hands, and you're about to see right now why hand washing with soap is so important when we're talking about pathogens or disease-causing agents. Soap is really effective at dissolving the outer coats and just taking those things apart and neutralizing them. So simple old soap is really going to be one of your best weapons against something like that. It's another reason your hands dry out when you wash them a lot, because the soap is actually dissolving the outsides of some of your skin cells, and that can result in dry skin. You're actually doing minor damage to your cells, but it's very minor. Your body's very good at repairing that sort of thing. So all along here, we should be continuing to crush up our mixture, smash it, mix it. At this point, the soap is doing most of the heavy lifting. Your hands aren't really doing as much of the work, even though you're still moving it around, but the soap is chemically opening up each and every one of those cells. Now at this point, you're going to want to get the piece of fabric that you have and at least one of your cups. I'm going to use a beaker here because we happen to have them. And you take your piece of fabric, whatever it is. It can be an old t-shirt, an old gym sock, little rag, anything. And you're going to want to put that over the top of your cup. Now, if you have more than one person available, this is a really great step to use a little teamwork. I'm going to have somebody hold the fabric around the cup and kind of press in a little bit so you have a little reservoir. Now this is going to turn into a filter for us. And this is going to catch all of the undissolved chunks and pieces of cell and all the other stuff that's floating around in here at the moment. And you're going to want to pour the contents of your bag out over your filter. Now this is going to be a really thick slurry of soap and cell pieces and uncrushed strawberry bits and all kinds of kind of sloppy looking this and that. Now, see down inside the beaker, I've already got some liquid draining in there. Now, I've just gathered my filter up, and you don't necessarily have to do this because this does not take a lot of liquid to get the DNA out. But if you want to, you can bunch it up, kind of give it a good squeeze, and kind of press most of that liquid on through there. Now, what we have inside our cup at the moment is salt, dish soap, and water, and all these crushed up pieces of cell. But there's also DNA in there, and the DNA is dissolved. Just like any other molecule can, DNA can dissolve. And if you've ever mixed salt and water together, you know that when something dissolves, you can't see it, but you know it's there because if you've ever taken a sip of that salty water, it tastes like salt. So we know that the DNA is still in there when it's dissolved. You just can't see it. So our next step, and this is probably the trickiest part of the whole thing, we are going to have to make the DNA undissolve from this solution. And this is a step that chemists call precipitation. Whenever you take something that has been dissolved and make it come out of solution, we say it is precipitated. This is where you're going to need your alcohol and another cup. Now, a smaller cup is better, but if you only have a big cup, it'll still work just the same. I'm going to do mine in a test tube because the results will be more clear to you on the camera. But again, if you only have a cup available or you have a small shot glass, something like that, that would be ideal as well. You can certainly use your whole solution. My recommendation would be take a little sample out of this, put it into another cup for this step, because this is where you can kind of experiment a little bit. If it doesn't work the first time, you can toy around with some of the variables in here, and I'll explain which ones are most likely to go wrong if it doesn't work on your first step. So I'm going to take a small sample of this. I'm going to suck it out with a little pipette, and I'm going to squirt it down into the bottom of my tube. I might get a little bit more. You can certainly pour yours, but keep in mind, this DNA, it's dissolved in here, but every single strand of DNA is almost 10 feet long, and it's fragile. So let's try not to beat it up physically a whole lot, because that can and will break some of those DNA molecules. You'll still get them to come out of solution, but they won't be quite as long. Now this is when you're going to want to go for your rubbing alcohol. Now I chilled my rubbing alcohol, but you don't necessarily have to. If your extraction does not work exactly as well as this one does, and actually I don't know that mine's going to work, we're going to find out. But if your extraction doesn't work right away, try putting your alcohol in the freezer for a little bit. Or as I've done, I just put it into a glass full of ice water here to get it a little cooler than the surrounding air temperature. Now I'm going to suck a little bit out, and I'm using 91% isopropyl alcohol. I'm going to suck a little bit out here. I'm going to add it to the test tube by kind of squirting it down the side. And the reason I'm squirting it down the side is even just the rush of rubbing alcohol hitting your sample in here can be enough to rough it up or break it up. Now, if you have a whole cup full of this, it won't matter as much because there's going to be quite a bit of DNA. So if I had just added the alcohol to this whole beaker, I probably could have just splashed it on in there. Now, what you're going to see here, and actually I can see the DNA in this sample already, you should have two layers. You have your alcohol layer on the top, and you have your mixture of cell pieces and soap and whatever else was in the bag on the bottom. DNA will dissolve in water, especially water that has soap and salt in it, but it will not dissolve in rubbing alcohol. So we've added this called rubbing alcohol because the DNA will come out of solution whenever it touches it. Now if you look really closely, you can already see that we've gotten a good DNA sample. You should see kind of like a slimy, bubbly blob inside this tube. It's the lighting good for that? You want me to move it?
You can see it a little bit. It just looks cloudy. They can probably see it better in person.
And if you tip it to let more of the rubbing alcohol, and again, be gentle because the DNA is fragile, tip it to let that rubbing alcohol mix in with it a little bit. When you straighten it up, you should see what kind of looks like, almost like a little ball of snot floating up at the top.
Kind of like gelatin.
It's like gelatin, yeah, it's jelly-like. This is your DNA sample. Now, for the next step, we're either going to use a toothpick or a skewer. I'm going to see if I can get it with my pipette here, which is not long enough. I didn't put enough in, so I'm going to use this little brush. Anything that has some fine edges, like a toothpick, will have little microscopic splinters.
Like a cotton swab if it's long.
You can dip that in there and give it a gentle twist, and the DNA should tangle up with it. There we go. And you can see it dragging up the side of my test tube right now.
Yeah, there it is. That little
really
blob.
thin fibers, and actually a big blob of it just rolled down there. That is actually your pure DNA sample. Now I'm going to let everyone who's working at home sort of catch up, work through. If you have questions, share them. I think Sarah's trying to answer as many as she can in real time if possible. If not, we'll try and answer your questions after the fact, or you can email them to us. But I'm going to go ahead and take a little sample of this and put it into one of these tiny little Eppendorf tubes, which I'm sure is something you don't have laying around the house. And I'm going to add a little bit more of my pure chilled alcohol to the top of this tube. This should be a very, very clear result here. And I'm going to cap the tube. I'm going to give it a little shake. And oh yeah, sure enough, there you can see the little gelatin blob starting to form at the top there. It takes a second to precipitate out a solution. And then I'm going to reach down in there with my pipette and see if I can't lift some of that out too. Oh yeah, look. That might be, oh, it just broke, but that might have been 1 or 2 single strands of DNA, and it is so thin and so tiny. It's amazing you can even see it. So I started a bag over here with some banana in it too, and generally that works just about as well as the strawberry, if not better. One of the reasons that we're using bananas and strawberries in addition to them being soft is a strawberry, I believe it has 96 copies of every strand of DNA in every single cell. To put that in perspective, a human body, every cell, or just about every cell, has 46 strands of DNA wound up into chromosomes. A strawberry has 96. It actually has 8 copies of each chromosome. And a banana has a similarly large number of copies of each chromosome. So there's a lot of DNA in each and every one of these cells that we're smashing up, which makes this a little more, little easier, a little more visual with these two fruits. But if you want to try this again and you don't have a banana or a strawberry available, anything, any living cell should work. You could take a piece of any plant. Plants are alive, they're comprised of cells. Any cells, you could break a little piece off an aloe plant. You could take a leaf off a house plant. You could go outside and get some grass. Actually, one of the things that works really, really well is anything that's actively growing and growing fast. So right now, the trees are starting to grow buds and release their little flowers. That stuff is growing very quickly. The picture that we looked at earlier here in the microscope is actually from an onion. And I tried a piece of onion and a piece of green pepper last night and got a really nice DNA sample out of it as well. So you can extract DNA from just about anything that's living, and that makes this cool experiment because it's very open-ended. You can actually go and test stuff and get your own data with this one later at home. It takes very, very simple stuff that's found around the house. Now, for those of you watching that notice some of the stuff we were using here, if you are having a little trouble working along with this with your cups or your glasses, or you would just like some of this simple science equipment type stuff, test tubes, eyedroppers, brushes, things that you might not be able to just go to the store, especially right now, and get, reach out to us. We have a kit that we've just made available that's going to have some science basics that'll have applications to just about all of the experiments that we're going to bring to you in this series. Tonight is step 1 of what is going to be at least a weekly online series of experiments that we're going to bring to you. And we kind of like your feedback on what we do next. So we have some ideas already, things that'll be fun to do at home and kind of different from the normal at-home science stuff. But if any of you out there have something you're really interested in, or have something that you've heard can be done, or even want to do a little digging around online and look for something that you'd like us to give a try, please email us, put it in the comments below, and we'll get back to you. We'll try and work in some of those ideas because we'd really like to do some stuff that you're really interested in. Also, on our YouTube page, we have a ton of videos. Some of them are just silly. Some of them you can work along with some other ideas for experiments to do while we're all tied up here. And if you go to our website, now that the weather's starting to turn nicer, we have just released a kit where you can build your own rocket launcher and launch rockets. And the rockets you build yourselves are just made out of more or less garbage waste, things that you would throw away. Empty water bottles and empty soda bottles. You're making a genuine rocket, and it can go pretty high up. I'm thinking we've logged them at least 70 or 80 feet so far. So, and if anyone has any questions, please reach out to us. We're here to answer. We're all going to get through this thing together. If there are any teachers out there watching anything that are looking for some interesting ideas or would like to collaborate on lesson plans, we are more than willing to help you with that sort of thing. So send us an email, info@outsidetheboxlabs.com, or check out our social media page. We're at OTB Labs, #OTB Labs.
The kits
Outside the
are
box.
under home kits.
Say again.
The kits are under home kits. So there's
Kits are under home kits on the website.
There's actually a DNA kit there too if you want the actual little test tubes and things like that. You want to order that and do it again with all the stuff.
All the specific stuff is in that kit, which is pretty cool. But again, this is something we'd really like to hear about your results from this. If you got a sample, if you didn't get a sample, if you're having trouble, we can probably help you walk through it again on your own time. And if you end up getting curious and want to try this with some other type of living thing, maybe you want to use a piece of a leaf or grass or a flower or any of the things we just discussed, we'd really be interested in hearing and seeing, if you want to take pictures, what your results are and what you're able to get. So stay in touch with us. We will be back next Thursday at the same time. We'll send everybody out a list of what we'll need and what we're going to be doing. We'll be looking forward to some of your suggestions on what we should do next week.
And hopefully without the technical difficulties.
Yeah,
for the first 10
I'd like
minutes.
to apologize for that.
Sorry, guys.
You know, this was day one for us. This is
First
the
day
first
getting it
time
set
we've
up.
streamed. We didn't see that
Live
coming.
stream, yeah.
Yeah, so that was
Little kink. Some little kinks in there.
a little bit of a surprise here. But we hope everyone had some fun. We hope we've given you something to work along with here at home, something you can take a little further on your own, and we will see you in a week, and hopefully we'll see you in person really, really soon.